Previously, you've learned about gram staining method as one of the techniques used for microbial characterization. In this session, you're going to learn another method of microbial characterization, which is biochemical testing. Obviously, techniques in microbial characterization are commonly used for microorganism identification, and this is really useful for several purposes. First of all, we can determine pathogens. For example, if infectious disease outbreaks happen, identifying microbes involved in the disease can help to ease or even eradicate the outbreak. Then, we can produce several products such as antibiotic or vitamin by isolating a certain strain of fermentative microorganisms. Isolating strains of microorganisms can also be useful to enhance the sensorial characteristics of a product. And lastly, microbial characterization can be used to compare biochemical activities of microorganisms, so we can use this for taxonomy purpose. In general, there are two methodologies that can be used for bacterial identification. The first one is genotyping method, such as using PCR or DNA sequencing to identify genetic materials of the microorganism. And the second one is phenotyping method, such as biochemical testing, which will be discussed in this session. Just as human beings possess a characteristic and specific set of fingerprints, microorganisms also have their own identifying biochemical characteristics. These so-called biochemical fingerprints are the properties controlled by the cell's enzymatic activity. The biochemical transformations that occur both outside and inside the cell are governed by biological catalysts called enzymes. And so, we can divide the biochemical activities of microorganisms into two groups. First, those with extracellular enzyme activities, such as starch, lipid, casein, and gelatin hydrolysis. And the second group are microorganisms with intracellular enzyme activities. So, we can distinguish microorganisms by identifying its ability to ferment carbohydrate, to produce hydrogen sulfide, and also to reduce nitrate or any other indicators. In this session, we will learn about carbohydrate utilization tests. This includes glucose metabolism using methyl red Fogg's Proskauer test, sucrose metabolism using 1% sucrose phenol red broth test, and also lactose metabolism using McConkey broth test, and then alternative sugar metabolism using Kligler's iron agar test, and also oxidative fermentation test. The MRVP tests are used to determine the ability of microorganisms to degrade and ferment carbohydrates with the production of acid and gas. The pH indicator methyl red has been found to be suitable to measure the concentration of hydrogen ions between pH 4.4, which is indicated by red color, and 6.0 with yellow color. Whereas in the VP test, glucose fermentation is indicated by the formation of a pinkish red-colored product. In SPRB tests, you are not only observing the change in color, but also presence of gas that will be visible as a bubble inside the inverted Durham tube. Cultures that are not capable of fermenting a carbohydrate substrate will not change the indicator, and the tubes will appear red. The lack of carbohydrate fermentation by some organisms should not be interpreted as absence of growth. The organism can use other nutrients in the medium as energy sources, and among these nutrients are peptones present in nutrient broth. So when this occurs, the phenol red turns to a deep red in the now basic medium. The pattern of sugars fermented may be unique to a particular genus, species, or strain of microorganisms. Lactose fermentation is one test that distinguishes between enteric and non-enteric bacteria, and the medium that commonly used to identify lactose fermenting microorganisms is McConkey broth. Similar with SPRB tests, in the McConkey broth, we also use Durham tubes to observe the presence of gas. Purple color in this test indicates there is no growth of lactose fermenting microorganism or negative result, whereas yellow color indicates a positive lactose fermentation. The Kia test is designed to differentiate among the different groups of the Enterobacteriaceae. This differentiation is made on the basis of differences in carbohydrate fermentation patterns and hydrogen sulfide production by the various groups of intestinal organisms. In this method, the slime is inoculated by means of a stab and streak procedure. This requires the insertion of a sterile streak needle from the base of the slant into the pot. Upon withdrawal of the needle, the slanted surface of the medium is streaked, and following incubation, you will determine the fermentative activities of the organisms. Red slant and yellow pot with or without gas production indicate glucose fermentation has occurred, while yellow slant and also pot with or without gas production indicate lactose and or sucrose fermentation has occurred. Whereas if the slant and pot are red, it means no carbohydrate fermentation has occurred. Lastly, only cultures of organisms capable of producing hydrogen sulfate will show an extensive blackening in the pot because of the precipitation of the insoluble ferrous sulfide. You can see more on how to interpret the result by studying this table. Finally, the last biochemical test that we will cover in this session is the OF test, or oxidative fermentative test. In this test, we step inoculate the medium and add sterile mineral oil into one of each pair of tubes, as you can see in this picture. Following the incubation, we can observe the result and interpret the biochemical activities that happen as a result of incubation. So that's the overview of the carbohydrate utilization test for microbial characterization. Now, please watch these videos to learn the real practical lab session. And don't forget to play the simulation on Labster, but after the live lecture session.